Thank you, and good afternoon, everyone. I'm Beatriz Urda, a PhD candidate at the Barcelona Supercomputing Center in Spain. And today I'm going to share with you a little story leading to disentangling the genetic and non-genetic origin of disease co-occurrences. So it's striking that 1 every 3 adults suffers from 2 or more diseases worldwide. And I'm sure that most of you already know cases like this, such as smoking increases the risk of developing lung cancer. But actually, there are many diseases that co-occur more than expected by chance, and these are often between seemingly disparate conditions. And we will call these disease pairs disease co-occurrences. And over the years, large-scale epidemiological networks have been built connecting these disease pairs that tend to co-occur in the same person. So these non-random patterns of disease co-occurrence suggest that there may be a biological explanation for them. Nonetheless, systematic methods have significantly recalled a small proportion of the disease co-occurrences or comorbidities, leading to the following question: do disease co-occurrences have a generalized molecular basis? This was the question of this very soon-to-be published manuscript entitled Patient Stratification Reveals the Molecular Basis of Disease Co-occurrences. And in it, what we did was to take a stratified gene expression and show that it substantially uncovers this disease. Specifically, what we did was to compile RNA-seq studies tackling human diseases and to derive disease similarity networks connecting diseases based on the similarities and dissimilarities of their gene expression profiles. And then, since patient-specific patterns have been observed in this phenomenon, we also took a stratified approach in which we divided diseases in groups of patients with a similar expression profile and inferred a disease stratified similarity network connecting both diseases and subgroups based on, again, the significant similarity of their expression profiles. And as you can see in green, both networks significantly recall a substantial portion of the co-occurrences. And in this manuscript, we also go over and provided insights into the mechanisms underlying this phenomenon in general, but also provided specific examples, for instance, going into the details and mechanistic observations for comorbidities that currently lack a biological explanation, and also were able to provide novel, potentially underdiagnosed co-occurrences for which there is robust molecular evidence, and everything is available in a web application that we implemented in which you can inspect diseases, their interrelations, and the molecular mechanisms underlying those. So as often happens, I think for most of you also, papers lead to other questions. And the thing is that transcriptomics is such a rich source of information that transcriptomic similarities behind disease could potentially reflect a wide range of factors, ranging from genetic to environmental influences. So this led us to try to disentangle this genetic and non-genetic origin underlying co-occurrences using as a proxy transcriptomics and genomic information. So for transcriptomic information, we use, again, RNA-seq studies for human diseases. We expanded around 3 times our data sets and reproduce the previous method to derive, again, networks connecting diseases and the diseases subgroups. And it was nice to see that we were able to reproduce our results with even improved recalls at the stratified level of around 80%. And then for genomic information, we used the UK Biobank, and there was a nice paper from Dong et al. That provided the pairs of diseases that co-occur in this population, and also the disease pairs sharing genomic components such as SNPs and other genomic components hierarchically inferred from SNPs, such as genes, protein-protein interactions, pathways, or significant genetic correlations. So the first thing that we did was to compare the transcriptomic and genomic contributions to disease co-occurrences. As you can see, SNPs in dark green recall a very low proportion of co-occurrences, around 3%, with increasing values for other genomic layers, and then until gene expression. And here, the most frequent scenario was unexplained co-occurrences, followed by transcriptomics. Nonetheless, when we consider the stratified approach, we see that transcriptomics is the molecular layer that explains that solely explains most of the co-occurrences, leading by unexplained disease. So if we put it on all together, the joint recall would be over 93%, leaving unexplained less than 7% of the co-occurrences between diseases. And as you all know, there are biological relationships between these components, and also the way in which the genomic layers have been derived hierarchically from SNPs. This led us to question, are there dependencies between them? And we took a directional dependence to measure this. And what we saw, mainly the main result, is that gene expression seems to be independent from the rest of the layers in terms of the disease that it can recover. But if we take a deeper look into the behind the scenes, what we see is that actually transcriptomics recalls most of the co-occurrences explained by the genomic layers, reaching from 77 to 95%. However, out of the transcriptomically explained disease, we see that around 60% are supported by genomics and have at least in one genomic layer. But there is a remaining 40% that have unknown genetic causes or non-genetic causes, which could be due to regulatory factors or environmental influences on gene expression. So at this point, what we can also do is to ask ourselves, how do diseases distribute in this molecular space of transcriptomics versus genomics? So in the y-axis, we have the co-occurrences explained by transcriptomics, and in the x-axis, the co-occurrences explained by genomics. So we will have in purple, those that are better explained by transcriptomics, and in yellow, those that are better explained by genomics. And this is what the molecular landscape of disease looks like. So mainly we see that most diseases are located in the first two quadrants, indicating that they are generally well captured by transcriptomics, but it exhibit a wide range of genomic contributions. And then we can go and look at the different scenarios that we can see here. So for instance, in gray, in the top side of the plot, we see diseases that are well captured by both omics, and these are generally diseases that have strong genetic influences and also considerable environmental factors. Then we could go down and see that there are diseases with important genetic susceptibilities, such as type 1 diabetes. And even there are some diseases, a few diseases that are better captured with by genomics. And here we could find, for instance, monogenic conditions. There are also some diseases that are like kind of in the middle. And an example that everyone understands very easily is obesity, which actually is due to an interplay of genetic and strong environmental influences. And then we find numerous diseases that are better explained by transcriptomics. And here we find most or almost all of the diseases in our data sets whose strongest risk factors are environmental. Some examples are post-traumatic stress disorder, smoking, or alcoholic liver disease. We also find most brain disorders here, bipolar schizophrenia. Interestingly, almost all neoplasms are also located in this region, and especially those that have almost non-genetic factors into it, such as colon cancer and infections which have a low heritability. So the summary from this plot would be that the relative contribution of transcriptomic and genomics seems to reconstruct disease etiology. And then we can see each of the points in greater detail. And for instance, here we have some examples. The first plot in purple is a post-traumatic stress disorder, which is mostly explained by transcriptomics on the top, and it's actually 5 times better explained than genomics. And then we have a wide range and particular scenarios that we can take a look. For instance, obesity here that has a more balanced contribution from the different layers, especially in genomics, at the molecular components, which is normal for complex diseases. And then, for instance, in green, an example of a monogenic disease that has under their co-occurrences a lot of contributions from, for instance, disease-associated genes. So if you recall from this plot, we saw that diseases tend to distribute based on their etiology. So this led us to want to systematically evaluate if there's a relationship between the genetic components of diseases and their co-occurrences. And to do so, we use the heritability, which basically measures the genetic component of diseases. And luckily for us, there was a nice paper from Westergaard that estimated large-scale heritability measurements from human diseases. The first question that we ask ourselves is whether or not genetic diseases tend to co-occur with other genetic diseases. And generally, the answer is no. We used a network theory concept, which is assortativity. I'm sure many of you are familiar with that. And what we observe is that diseases do not tend to co-occur based on their heritability values, except when they share SNPs in dark purple. So the SNPs seem to have like a key role in linking diseases based on heritability. However, what we do find is that these genetic diseases, even though they do not tend to co-occur with each other, they tend to have genetically driven disease. And this is true for heritable diseases when they link with other heritable diseases, but also when they link with diseases that are not highly heritable. And the way in which we reach to this conclusion is by computing the correlation between disease heritability and the proportion of co-occurrences that are explained by genomics. So as you can see, when we do this for genomics, we see a significant positive correlation. However, this is not the case for gene expression. Gene expression seems to recall comorbidities regardless of heritability, which shows that it is able to recover genetic factors, but also environmental influences or regulatory influences on gene expression. And we also try to do an indirect way of measuring if actually we are recapitulating some regulatory elements beyond DNA sequence. So we computed this correlation, but for the relative proportion of transcriptomics and genomics. And indeed, we saw a significant negative correlation, indicating that at least that proportion of the transcriptomic disease might be explained by factors beyond DNA sequence. And this is also in line with observations made with spousal heritability, which is a proxy for the environmental component of diseases. So to conclude, what we saw is that approximately 60% of transcriptomic disease are supported by genomic components, whereas the remaining 40% are not explained by the known genomic layers, suggesting contributions from regulatory or environmental mechanisms of own expression. We also saw that that the relative contributions of transcriptomics and genomics reconstructs disease etiology, that genetic diseases do not tend to co-occur with genetic diseases except when sharing SNPs. However, they tend to have genetically driven co-occurrences even with lowly heritable diseases. And on the contrary, transcriptomics explains disease regardless of heritability, and at least partly due to non-heritable mechanisms. And finally, as a high-level conclusion, that by integrating genomic and transcriptomic data provides a near complete coverage of disease among the analyzed diseases, with a considerable portion likely rooted in factors beyond DNA sequence and therefore potentially modifiable. So finally, I would like to thank everyone in our team, and especially my supervisors, Alfonso and Davide. And I've included here some QRs for the web application and for socials, in which we are going to be sharing soon the soon-to-be published manuscript and the preprint. Thank you. I'm happy to take questions.
Hi, thank you very much. I have probably an obvious question. How did you deal with the tissue level differences between the transcriptomics? How did you correct for that? I assume that your RNA-seq data came from different tissues, depending on the disease, or the main manifestation of the disease.
Exactly. Thank you for the question. It is a very good question indeed. It was something that worried us from the beginning a lot, and we have made a lot of tests in that regard. What we saw is that actually there seems to be enough systemic effects to observe co-occurrences, even when we are comparing diseases from different tissues. We are correcting for the tissue effect because we are considering similarities between differential expression profiles. So for each disease, we have patients and controls, so we are able to assess that. But it was surprising to see that there's enough systemic effects to observe co-occurrences even between conditions that are in different tissues. And indeed, I think that it's just like 25% of the interactions that are within disease categories. So most of the similarities that we find are across tissues and in core molecular processes, such as, for instance, chronic inflammation is the main factor driving almost all of the disease co-occurrences, which is known to be able to have like, a systemic effect in many cases.
Okay, so did I get it right that you removed kind of the differentially expressed genes that were unique to a certain tissue, or how can I understand?
No, what we did was to, since we are using differential expression profiles to compute the similarities, we are already having like the cases and the controls from a given tissue. But then we are not removing tissue-specific genes. And we did some analysis to see if by removing the tissue effect, we see any differences. But what we saw is that what's driving the similarities between diseases are systemic effects.
Okay,
thank you.
Thank you. Very interesting presentation. And I have noticed that on the top right corner, when you compare the various this one, yes, you found these both 100% of the captured by genomics and transcriptomics. There was ulcerative colitis, which is one of the diseases which I am researching. How you would interpret that it has so much environmental and genetic component in this case?
Ulcerative colitis is actually highly heritable, and but there are many environmental factors that affect this, such as lifestyle, diet, so on and so forth. So what we see is that transcriptomics captures not only environmental regulatory factors, but also genetic effects on gene expression. So that's why maybe transcriptomics is also able to capture that, because it also captured the reflection of the genetic alterations that may happen in ulcerative colitis. Whereas this is not the case for diseases such as, for instance, post-traumatic stress disorder, which is also captured at a high level at transcriptomics, but not at with the genomic component. But indeed, we also can go deeper and explore specific diseases. And for instance, we can find that there are some interactions that are captured with both omics, but then some of them are differentially captured. And but with ulcerative colitis, it works really well. It happens the same with asthma, which is also highly genetic, heritability of almost 90% in children, and again, highly captured by both omics.
Thank you.
Thank you.
Hi, thank you for the talk. Just a follow-up question. How do you handle, for instance, a fact of the disease like inflammation? If you have ulcerative colitis, IBD, neurodegenerative disorder, after inflammation, you see like an impact. Is then the RNA picking up the inflammation? That's why you see the differentiation. How do you handle that?
Exactly. So the first thing that we did is to see significant similarity between the differential expression profiles, and this is a number and a p-value, right? But then we can go and see, okay, what are the genes or the pathways that are altered in common and in the same direction? And we have found and described many examples in which inflammation seems to be like the strongest signal, not the only one, but the strongest signal underlying the vast majority of comorbidities, but also in specific comorbidities being the most important factor in many pairs that are from different categories. Another example of that would be the relationship between asthma and Parkinson's disease. And for instance, in that case, we've done like a detailed explanation on why the immune system is driving that. So it would be like going after that to the results, which are lots of results, and try to derive mechanistic explanations from that. Thank you.
Okay, well, I will invite the other questions to be posed and asked during the feedback. I mean, thanks to the speakers, we are on time, which is already nice.